everyone. So today we will start with the eukaryotic cell. Yesterday we did prokaryotic cell. I made the diagram for you all of prokaryotic cell. Today we are going to start with eukaryotic cell. So if we see the eukaryotic cell is of two types. Eukaryotic cell it can be either a plant cell or an animal cell. It can be either an animal cell or a plant cell. So one by one, we will take both the types. Just now, I'll take a first the animal cell. The shape of the animal cell is mostly oval. Single layered, that means only cell membrane is present. The nucleus is centrally located. Like this. We have a densely located body in this, a dense body. Then we have this, which is chromatin fiber. Then besides this, to the nucleus is connected the and network of endoplasmic reticulum. This way. Some of the endoplasmic reticulum, they have on their boundary the ribosomes attached to them, like this. Along with this, we have here the Golgi body. Then in the plant cell, the vacuoles are many and small in size. Many vacuoles are there and they are small in size. Along with this, we have lysosomes, which are filled with hydrolytic enzymes. we have a number of mitochondria where the inner wall has infoldings which are known as cristae. This way. And we can make one more somewhere. Along with this, we have the only structure, the structure which is present in animal cell, that is centriole. So, two centrioles which are placed together, one is this direction, another is in this direction, and together they form the body centrosome. So these are two centrioles which are there, and we have cytoskeleton also. Cytoskeleton. This way we can make some cytoskeleton also. And besides this, we have small vacuoles, and these we have made. Now, if we label it, how will we label it? This is cytoskeleton. This is plasma membrane. This is mitochondria. These are Golgi complex, Golgi bodies. Then this is because it has, we can make some endoplasmic reticulum like this also placed here and there. Some of them are having the ribosomes on them. Some are without the ribosomes. So here, these are rough endoplasmic reticulum, RER, rough endoplasmic reticulum. Then we can make this, these are lysosomes. These are vacuoles. Then this is cytoplasm. And some ribosomes also we can make. Ribosomes, which are the protein factories of the cell. So most of the labeling is done. Now what is left is the nucleus part. So this is the nuclear membrane. And then these are the pores, nuclear pore. Then inside this substance, which is there in the nucleus, this substance, the ground substance inside the nucleus is nucleoplasm. This dark body is nucleolus. And this is chromatin fiber. So this is the labeling of the entire animal cell which I have drawn for you. And it includes all the organelles which are present in the animal cell. I think this is clear to you. Yes, beta. One, I have forgot to mark this one. This is SER, smooth endoplasmic reticulum, which has no ribosomes on. So this is smooth endoplasmic reticulum, and the complete marking I have done, complete labeling I have done. So I think this is clear to you, an animal cell, is it? Yes, beta, is it clear to you? Yes. Shall I switch over to the next cell, which is plant cell? Okay. I'm getting the response that it is clear. Yes, I'm getting the response. Now I'll switch over to the next cell, which is plant cell. Plant cell has a double layer, so usually it is made like this. In some cases, it is made in polygonal structure. Somewhere it is made in hexagonal. Somewhere just a rectangle-like structure. So it can be made anyway, but in both your book, NCERT books, you will find it out like this. This is how you make it. And here the center line, the membrane which is present in the center between the two is middle lamina. This is middle lamina. Now, this is a characteristic property of a plant cell, that it has few but large vacuoles. So that large vacuole we can make on one side, a large vacuole which is present in the plant cell of the shape. Okay. Now, besides this, what we have in the plant cell is, because of a big vacuole, the nucleus is not centrally located, it gets displaced to one side. So we will have the nucleus like this here. Like this. Besides this, the same way, what we have in plant cell is the endoplasmic reticulum. So endoplasmic reticulum needs to be made. Like this. And we can make some over here also, this side. Here. And we can make ribosomes on it. like this. Then we have the Golgi complex, which is smaller in size and not placed the way it is placed in the animal cell. And in case of plant cell, the dictyosomes are smaller in size, and these dictyosomes, which are dispersed and smaller in size, are these Golgi complex are known as dictyosomes. Besides this, we have mitochondria. This way, the mitochondria is present. The number of mitochondria depends on the rate of metabolism of a cell. Then we have chloroplast. We have the plastids named chloroplast. Chloroplast has a number of thylakoids which are placed one over the another. Then we have stroma lamella, a number of thylakoids here like this. Okay, so I made the chloroplast also. Now, besides this chloroplast, lysosomes are present, but lysosomes in this case are few in number. Only few lysosomes are present. So I'll be making only 2 lysosomes here like this. The lysosomes are present, which are few in number. Besides the chloroplast and this, we have some microbodies. The microbodies are present. I'm making it dark. These microbodies. Then we have some ribosomes also. Then we have the cytoskeleton. We have the cytoskeleton. So this is what we have in our plant cell. And this plant cell, the labeling, I'm going to do it for you. This is a vacuole. This complete is a vacuole. And this vacuole is filled with cell sap. Cell sap is there. Then this is cytoskeleton. This is cell wall. The middle one is middle lamella. The inner one is cell membrane. Then we have this mitochondria. Inside we have the cytoplasm. Then these are lysosomes. Then we have the chloroplast. This is dictyosome. Dictyosome in case of plant cell is the Golgi body in case of plant cell is known as dictyosomes. Then we have these ribosomes. No, microbodies. These are microbodies. The microbodies are peroxisomes or spherosomes. These are the microbodies, spherosomes and peroxisomes. And also peroxisomes. These are the microbodies. Then we have the ribosomes, which are protein factories of the cell. Then we have this rough endoplasmic reticulum, R, E, R, and we have the smooth endoplasmic reticulum, SER. Then besides this, we have this nuclear membrane, NM. We have this nuclear pore. We have this nucleolus, the nucleoplasm, the chromatin fiber, And all this from nuclear pore to the chromatin fiber, these are the various components of nucleus. These are various components of nucleus. And the diagram is incomplete unless you write what is it about. So a plant cell, which is a eukaryotic cell. So this is the plant cell which I have made for you. And I think you are able to understand what I have made. This cell sap, it consists of the cell sap which is present inside the vacuole. It consists of minerals, water and minerals, H2O plus minerals. This consists of water and minerals. So this is cell sap and this is the diagram of plant cell. I think you are able to understand it. Anything which you could not understand in this or anything about which you are not clear, just let me know. Yes, beta. Yes, I'm getting the message. Understood. Yes. Anything which you want me to tell you again? Yes, bache. I think the diagram is clear to you all. Okay. So I'm getting everything is clear, ma'am. So now let's, because we have just, I think, 7 minutes with us. So in that 7 minutes, we will do the difference between plant cell and animal cell. So the difference between plant cell and animal cell, the first difference is the presence of cell wall. Cell wall is present here. And cell wall is present. Whereas in this case, the cell wall is absent. This is the first difference. The second difference between the two is the plastids. Plastids means, what are plastids? There are three types of plastids: chloroplast, chromoplast, and the third one is leucoplast. All these three plastids are present in plant cell, whereas all these plastids are absent in animal cell. Another point of difference is about the vacuoles. Vacuoles are large and few. Whereas in case of animal cell, the vacuoles are small and many in number. Vacuoles are small and many in number. Lysosomes are few. Lysosomes are few in this case. Whereas in case of animal cells, the lysosomes are many. This is the fourth point of difference. We come to the next point of difference, which is about the centriole. Centriole is absent. In plant cell, whereas the centriole is present in animal cell. Okay. Now, any other difference which I'm forgetting and you remember? Yes. Yes, beta. These are the main 5 differences which I have told you between the plant cell and the animal cells. So one more difference you can write, that the nucleus is pushed towards one side in case of plant cell and it is centrally located. nucleus pushed towards case nucleus is centrally located. This is the difference. One more difference we have come to know. So this is the next difference between the two. And I think these are sufficient differences between the plant cells and the animal cells. Okay, so rest, if you remember any other difference, just let me know. Yes. Okay. So beta, next topic which I would like to start, I'll give you just the general introduction of that topic. And we will do it properly tomorrow, and that is plasma. Now, what is that? So plasma membrane. This plasma membrane is outermost membrane in animal cell. Outermost in animal cell, whereas second in plant cells. This is thin. First of all, I'll write this is living. This is a living membrane. This is thin. This is flexible. And one term which I would like to use for it, this is selectively permeable. Also. See, there are few terms like selectively permeable, semi-permeable, and impermeable. These are the three words, three terms which are quite often used, in which the nothing is allowed. Okay, then which allows everything to pass through it. So cell wall is fully permeable. It allows everything to pass through it. So this impermeable, which doesn't allow anything to pass through it. Semi-permeable, which allow few things depending on the size of the pore, which is, but